Good morning. My name is Marcela Souza. I'm a project manager at Ghent University, and one of the projects I'm working on is the ALG-AD project. And today I'll be talking about the results we gathered in the project about growing microalgae on plant-based digestate, the Belgian pilot site. So this pilot site is being operated by both InnoLab, which is an AD consultancy agency, and Ghent University, where I work. So I just want to start with a brief introduction on the ALG-AD project, where we propose a circular solution for the nutrient surplus challenge in Northwest Europe. So Northwest Europe is a very densely populated area with high amount of activities of agriculture and animal husbandry. And these activities generate a lot of waste, which is organic waste, that can be processed through anaerobic digestion into two streams, so biogas, which will be used for production of energy and heat, and digestate, which is the focus of this project. So digestate is a nutrient-rich stream that is currently being used as fertilizer. However, due to nutrient surplus problems, as I mentioned in the beginning, you have in Northwest Europe several areas where the soil is so rich in nutrients that if you apply too much, it will run off to water bodies and cause eutrophication problems. So what we want to do with this project is to use this surplus digestate that cannot be applied to the land as a nutrient source for microalgae cultivation. Then, once we have the biomass grown on digestate, which is protein rich, we process it into animal feed. And in that way, we can create a circular solution for this nutrient surplus in Northwest Europe. However, using digestate for growing algae, it's not a simple process, and it has a lot of challenges. And this can be summarized in four main challenges, which is suspended particles, high viscosity, dark color, and high ammonium concentration. And for the dark color, I have an example here in the picture. So you can see that even when digestate is diluted at 20%, it's still very dark. So only when we go to 10 or 5% concentration, which are very low, you start to see the transparency happening. So how is the Belgian pilot site dealing with these challenges? So I'll first start with the suspended particles, and for that I'll briefly explain what we do in the Belgian pilot site. So we get the liquid fraction of digestate from an AD plant in Belgium. This is further filtered in a 10 micrometer pore size filtration unit, which we call paper filtration, but actually is a cloth bag with a pore size of 10 micrometer. So this removes most of the suspended particles. And then this filtered system is fed to our reactors, which are situated at InnoLab. And this is different from the other pilot sites. So the two other pilot sites in the UK and France, they're focusing on using smaller pore size filters. So they use microfiltration and even smaller for the French pilot site. So we wanted to test how the paper filtration is comparing with a microfiltration. And this was not done in pilot scale, so this experiment was done in lab scale. And we have the green bars with the paper filter digestate at 10 micrometer pore size. And in blue, you have the microfilter digestate at 0.1 micrometer. So we compare these in different concentrations, and you can see that mainly at the lower size concentration, so 2.5 and 5% digestate concentration, the paper filter digestate gives a better growth than when using the microfilter digestate. And this result is important because, as I will show in the next slides, we are mainly using this reduced digestate concentration, so 5, 2.5 and even less. And why is paper filtration giving better results? So we have noticed that it maintains more nutrients. Of course, it has a smaller pore size. So some of the suspended particles actually remain in the digestate, and these are sources of nutrients. Also, the paper filtration because it uses 10 micrometer, which is bigger than bacteria size, it maintains the benefic bacteria that are actually helping to make the nutrients more bioavailable for the microalgae cells. And also on a technical economic perspective, the paper filtration has a lower energy expenditure due to its higher pore size, and so it has lower cost when compared to the other filtration units with lower pore size, smaller pore size, because there you need to apply pressure. In our paper filtration unit, gravity is the driving force for the filtration. However, it doesn't come without disadvantages. So there is a possible presence of pathogens. So the same way that it allows the good bacteria to remain in the system, it can also allow pathogens to stay in the system. However, we have tested this in the project, and this was not a cause of concern. And so paper filtration also maintains most of the color of the digestate. If you have seen the previous webinar, Florian, she presented some results with microfiltration and ultrafiltration, and there you see most color removal. And here with paper filtration, because some suspended particles still are in the system, you still have a dark color. However, this is not a problem, as I'll show in the next slides, because we dilute the stream. So for the other three problems, the high viscosity, the dark color, and the high ammonium concentration, we deal with them basically with the same solutions, which is dilution and fed batch. And I have here a graph from the Belgian pilot site to show how this is effective. So what we have here in blue is the biomass growth, and you can see the unit in the secondary axis to the right. And in orange, you have the nitrogen concentration coming from ammonia, and this is on the primary axis to the left. So what we did was we started with around 50 milligrams per liter of ammonia, which is lower than the toxicity level, which is around 100. We let it consume. So for about 6 days, the cells were growing, the nitrogen was being consumed. Then we fed the reactor again. We let it consume again, and then for 10 days it was consuming, and then we fed it again with a higher concentration, and then again it went down for the next 10 days. And you can see if you follow the blue curve, we were able to reach exponential growth. So this means that there was no nutrient lacking in the system. The cells were growing very nicely. And at the end, we fed 5% digestate to the system, which is around 150 milligrams per liter of nitrogen coming from ammonia. And we were able to reach a final concentration of 1.7 grams per liter. So no toxicity effects were seen, not even by light penetration problems or because of ammonia toxicity. So we also have a vertical system at InnoLab. So those results that I showed in the previous slide were for the horizontal reactor, but we also have a vertical reactor. So the horizontal is a commercial one by ALG-AD, and you will see in the next presentation more about this system. And the vertical reactor was designed by the ALG-AD team at Swansea University as a low-cost system. And so as I showed you before, with the horizontal system, we were able to reach a final concentration of biomass of 1.7 grams per liter, and the productivity was around 0.1 grams per liter per day. When operating the vertical reactor with the exact same conditions, so similar inoculum, similar feeding strategy, we were only able to reach 0.7 grams per liter with a productivity of 0.03 grams per liter per day. And as you can see in the slide, in the right side graph, the 0.7 was really the maximum that we could reach. We tried to operate the system for 10 more days, and there was plenty of nutrients available in the medium, and the algae did not grow. And this is mainly because, as you can see in the picture, the horizontal reactor has much larger tubes with a larger diameter than the horizontal reactor. So the lower results obtained with the vertical reactor are less because it's a vertical reactor against a horizontal reactor, and most because of the size of the tube. So with larger tubes, you get more light penetration problems, you get more self-shading by the cells, and you have a lower density that you can reach before having these light penetration problems. So just to be fair with the system, we did not operate this that much because we are getting better results with the horizontal. However, the UK team is operating the vertical reactor, and they were able to match our productivity. So they get quite high productivities with the vertical reactor. However, their concentrations are still much lower than ours, and they usually don't get more than 0.7, 0.8 grams per liter. Usually is even lower. So what are the next steps for the Belgian pilot? We want to optimize inoculum. So you see that we have modeled the system with the data that we have gathered so far. And in this graph, you see different percentages of inoculum and what the biomass production on grams per day we would get with these. So right now we are operating at around 20% inoculum. And in the graph, you see that if we go higher to 50% of inoculum, we might get higher productivities. Of course, this doesn't come without its disadvantages. So if you only renew 50% of your medium, it might be that you will get some toxic elements being accumulated in your system, which may cause the culture to crash. So we will see, and we have to find a balance between media renovation and a higher inoculum that will give us higher productivities. We also want to optimize the feeding regime. We got a good productivity, but we think that if we optimize it better and keep the nitrogen levels lower, we might be able to get even higher productivities. And most importantly, the way we did this feeding, we usually get around 20 milligrams per milligrams of nitrogen per liter at the end of the cultivation, which is higher than what the legislation allows for dischargeable limits. So right now we cannot discharge this water. And the idea is to get the system to also have clear water at the end that you can actually discharge. And for that, we have to optimize how much nutrients we give to the system. And finally, we want to recirculate the water, because right now, as I told you, we are using a very diluted system. So basically the whole volume of the reactor is clean water. And if we can recirculate this water after harvesting, so we take out the cells and the remaining medium, we just put it back into the reactor, we will reduce the environmental footprint of the process, which is the goal of the ALG-AD project. So thank you very much. I hope everything was clear, and if not, I'm happy to answer some questions. Thank you.